Let me go on to my talk, and the topic is treatment of microvillus inclusion disease, a personal journey. And the reason for this talk is really that I feel that so many things that relate to rare diseases are personal, starting from being a personal problem for the affected kids and a problem for the families and problems for everything, and leading to the fact that whatever is done for rare diseases these days is done by the individuals and done by the passion, by the curiosity, and by everything that makes us really just people and individuals working with the disease. So, and I'm going to try to share my journey and hopefully get somewhere. So this would be the first couple of slides, the introductory slides about myself. My name is Dmitry Kravtsov, and our company is Vanessa Research Incorporated, and we're local. So the headquarters are located in Hamden, and it's on Sherman Avenue, not too far from the Quinnipiac main campus. And we started in 2015, and our primary focus is on the development of treatment for the exceedingly rare disease called Microvillus disease, the MVID. And this slide just repeats what Carolyn has already said, but please feel free to read from it. But the point is that throughout my career, I was a modeler. And this is what I've been taught. I always had great mentors, and I was very fortunate to have excellent mentors in science. And the accuracy of the model and the specificity of the model is the key to any discovery. So I've been taught very well to make accurate tissue-specific models, and that has paid off really, really well throughout my scientific career. And it took our team at Yale about 4 years to from start to finish with the microvillus inclusion disease. And we started off with just as a logical continuation of our previous work with the myosin and trafficking in the intestinal cells. And it's culminated with a novel view and a novel perspective on the disease, which gave a book treatment. 5 slides exactly. What is microvillus inclusion disease? It's a rare genetic disease. And the affected gene is the gene of the myosin 5B, which is an intracellular actin-based motor protein. Disease affects just about 350 people worldwide, and that's the number of reported cases. How many of the cases go unreported is really unclear now because the diagnostics is very complex. The first step that lets a doctor, a treating physician, to suspect the disease is the electron microscopy of intestinal biopsies. So that's so far away from being like a primary and the first thing to consider that we simply don't know how many kids die with diagnosis of acute gastroenteritis without ever being diagnosed with the proper genetic congenital enteropathy. Right now, there is no treatment. And since the symptom and the main symptom of the disease is an incurable diarrhea, where kids can lose up to the third of their body mass per day with stool, it's currently managed by the parenteral nutrition when the fluid, electrolytes, and the nutrients have been replaced intravenously in the attempt to bypass the digestive tract. And life expectancy for patients with microvillus inclusion is hovers around 4.5 years right now. So the terrible thing about parenteral nutrition is that it's a wonderful short-term solution. So if you administer to somebody who has acute severe diarrhea, it's a lifesaver. It's wonderful. It's great. It's great if you continue it for days, maybe weeks. But if it's months, it becomes a problem because it's not free of complications and things like sepsis and the wound site infection, thrombosis start affecting the patients. But really, really terrible thing is that it takes about 4 or 5 years for TPM to really heal the liver. So it's inducing the cirrhosis, and eventually the suggested standard care right now is the combined liver and bowel transplantation. So, and Microvillus inclusion disease got its name from a very, very characteristic intracellular inclusions that are seen in the intestinal cells. And this is the inclusion that you see at the far side. Right there. So this is a very typical inclusion that gave the name to it. So what is the difficulty? What's going on with the Microvillus inclusion disease? What's the problem? We know that this is a diarrhea. We know that it's a genetic. It manifests soon after birth, and if the patient is more than about 2.5 months of age, it's guaranteed that whatever diarrhea happens, it's not an MVID because it's confirmed, it's directly associated with the genetics and the genes of MVID, the only gene that has been proven. That's known. MVID is an active motor. There is absolutely no link. There's nothing that connects intracellular motors to the ion transport to the secretion. The link is indirect. So where is the link? There are hypotheses. There's things that we can consider. There's many guesses, but nobody knows for sure why and how the motor, which just simply moves things inside the cell, when it's absent, manifests as a tremendous and severe diarrhea. And that was actually the quest that we worked on back in 2012. So let's start with what's known about the disease. It's a known, well-characterized secretory diarrhea. So these two short videos kind of explain what is wrong with the how the secretory diarrhea happens. And here we have a healthy intestine, and this is the slice of the intestinal wall looking from the body side into the opening of the intestine, the lumen. And on the right we have the Microvillus inclusion disease, showing some of the morphological abnormalities that are typically seen. Now, normally, everyone's intestine is handling about 2.5 gallons of fluid daily. This is not only what we drink and eat, this is also what body contributes into the intestine to make it function properly. So it's the secretion from all the glands, the salivary glands, stomach secretion, liver, pancreatic juice, and what intestine produces into it to make the digestion and absorption a seamless process. And these 2.5 gallons are efficiently handled by the normal absorption. So the flow of blue bubbles from left to right is representative of the incoming fluids. And then the absorption takes place by the structures called villi, while secretion is taking place from the little intestinal glands called creeks. And what we try to show here is that most of the incoming fluid is being efficiently absorbed into the body by the small intestine and the large intestine. And in the Microvillus inclusion disease, the process is very different. There's virtually no absorption happening in the MVID intestine. And then secretion is increased. So the intestine is not absorbing anything of this 2.5 gallons. And it's contributing more than the healthy intestine. Thus the diarrhea. But why is it? So what's wrong? Why the motor? And how is the motor linked to the diarrhea? Nobody knows. And the current concept focuses on the intracellular traffic and polarity disorders. Now, and these are 3 intestinal cells, 3 enterocytes. And this is the immunofluorescent flagellum that I did back in the Yale days. So every cell has a nucleus, and then intestinal epithelium is polarized. So it has a very strict division of functions between the top part of the cell and the sides and the bottom of the cell. This is where absorption takes place. This is the top part, and this is where the proper absorptive protein machinery is located. So this is a feature of the mature enterocyte. If you look at the MVID, cell, that you can clearly see that absolutely no green is visible at the top of the cell, and green is what is doing the absorption. So this is the answer. There is no absorption because there is nothing that can efficiently handle and take things into the body. But, interestingly enough, the normal, immature intestinal cell looks remarkably similar to the Microvillus inclusion disease cell, or vice versa, that the MVID cells looking just like the immature normal enterocytes. So when we found that out, we said, wow, interesting. Is that really that? Okay, so the problem is it's not just the trafficking. It's actually that the fact that the cell remains permanently immature. It can't absorb, it can't secrete. And if every single cell in the intestine is only capable of secretion, then again we want to do diarrhea. So this is the 4.5 gallons lost each day with stool. This is up to 500 cc's per kilo per day of the volume loss with children. Okay. When we realized that, then here came another question. On the left is the current model of the myosin 5B dependent trafficking in the enterocytes. So you have a multitude of different linkers, components, and facilitators of trafficking, and things that move ion channels, things that dictate where they must go, what is the proper place, and what is the proper trafficking pathway. Based on this picture, it's very, very difficult to come up with a treatment that is efficient and specific, because there is no known molecules that individually affect any of these components specifically in the intestinal cell. But if you think of the defective maturation, so it's a step up from the intracellular problem. It's a step up to the whole organ problem. And actually there's quite a few drugs known that do affect how cells mature in the body. So from the treatment perspective, the concept of defective maturation sounds like a shortcut to the treatment, because if you just simply make this cell to look like that by forcing it to mature by flipping the switch that is missing in the case of myosin deficiency, all of a sudden you get a chance to turn the intestine back into normal state. That was about the time when our paper was getting ready to publish. And this was the idea that really started to kind of to grow. Why don't we try to treat it? Why don't we try to come up with something that will specifically induce the maturation of the intestinal cells? So it was literally a critical point, because I was weighing and deciding, do you want to remain in science and just become like, I apply for an NIH grant and keep studying the disease and getting it into more and more details? Is we validated and confirmed our hypothesis. But it has, even now, it has way more questions than answers, and it must be followed up and verified and re-verified and validated. Or I say, okay, this is good enough. We should try to treat it, and we should try to come up with something that has a potential to affect. So, bottom line is, that's where my heart was. This is what I wanted to do. And again, coming back to personal, it's not the smallest part in my decision that I really wanted to follow up the disease from the start to the finish, from the idea that we came up with the way to treat it. So we want to finish it and make the treatment real. And this is when I decided to join Vanessa Research. This is the end of science slides and the start of business. And this is the wonderful business slide right here. So I plunged into the cold water of drug development. And it's represented here as a flower. Okay? And every petal in this flower is actually a huge division, a huge endeavor. Because when you do the drug development, you have to have the preclinical research. And it's not just simply having a nice idea. It's just actually having the data and a well-documented data for every single experiment and everything you do. It's manufacturing. How do you make the drug? So yes, we have the idea. Yes, we have the candidate molecules. Who's going to make it? Who has the certificate and the license to make it so that it can be given to the patients? Where are the supplies for the drug? Now, we've made it, what about the clinical data? We need the data from test trials and tests in patients to really go ahead with the approvals. Okay, fine. What is the distribution? How are we going to ship the drugs to the hospitals and to the patients? What are the export-import permissions? In every country, it's individual. How about the FDA or the EMA, the European analog of the FDA? How about the intellectual property and legal coverage for what we do? So, on the right, for the major pharma companies, land is kind of cultivated. They've been doing it for years, some of them for over hundreds of years. I think that Merck is more than 100 years old company. It's trying to be wonderful. But for a drug development startup, we're on our own. We have two opportunities: we either grow or we die. And that's how a typical startups would look like if you compare them, if you continue the analogy. So then the mission, if this is so challenging and so difficult, so what keeps us running? Why are we still doing it? First of all, again, it has become my personal goal and my personal challenge to treat MVID. And I don't want to stop up until the treatment is out there and the treatment is available. The second is that the road is really interesting in its own, and not a single thing that we do is done in the silo. So we speak, we consult, we communicate, we interact, and through these interactions, we find many other possibilities that we can, and many other new goals that we pursue. So actually, the goal becomes just the next step, and we aim for the next step forward. And the process is really interesting, and I don't regret any single moment that I really decided to join the business side of the drug development and then continue with actually making the drug. And through that, and through the company, and through our interactions with people, patients, families, doctors, and everything else, I realized that how people live in silo, and there is a doctor silo, there is a scientist silo. When I was back at in the lab at Yale, you know what a patient was to me? It was a box of slides. It's just the slides which have like a tissue cut, a slice of tissue that I label, stain, get my data, and I get like a beautiful picture that is worth of scientific publication. And when I'm a doctor, my patient is one of the 23 or 24 people that are kind of on my list today that I have to see, talk to, and make records and really just treat, sign out, whatever. And the patient side is just like it's usually completely anti-doctor, anti-scientist, because the doctors are like kids in white coats who don't know anything about my kids, and I'm a father of two, so I know what I'm talking about, and my wife is even worse than I am in this regard, because whenever we take our kids to the doctor, she's never happy, so it's just like it's always something on there. But at the end, like connecting it and communicating is really important, and this is what the organizations and the rare disease support and everybody else should be doing. This is what we're all here today for. And through that, one of our primary missions has become to educate people regarding the microvillus. And it's not only the families and the patients that we're educating. My pediatrician has never heard of microvillus inclusion disease, ever. And the guy is practicing for 25 years. He's a wonderful pediatrician. He's never heard of it. So it's the doctors, scientists, and whoever else is in the disease that we're trying to address with our educational platform. Become the new standard of care. If we have a treatment, it's not a cure, it's a treatment right now. But still, it completely liberates patients of the parenteral nutrition. So there is no need for the 24/7 IV line in the kid. They don't live with a backpack that has the IV packet. It's a good thing. And if they don't need the intestinal transportation to live, it's even better. And we want to increase the awareness of the disease worldwide. There are many aspects to this. It's just some of them are very generous as to thinking of if doctors know more about disease, starting from students. You just hope, like if you guys hear it from me today and then remember it throughout your life, maybe when you encounter a patient with diarrhea, you would think of the MVID. If it's a baby who's like 3 weeks of age or 2 weeks of age with incurable diarrhea, well, nobody thinks of it. Maybe you will. I don't know. I hope you will. It's good for us because increasing the awareness increases our target patient population, which is wonderful for our trials. For instance, we need patients. We're going to be recruiting once the protocols are formal. We need patients on all in the lab. So this is a great thing. And if you are interested in reading more about MVID, please feel free to visit mvid.vanessaresearch.com. This is a free educational resource about the disease. And we have animations in there, we have text explaining it. We have a collection of scientific papers dealing with microvillus disease, and we're periodically updated with more information. So again, if you're interested in the disease, please feel free to read more about it. And I think it's time to say my thank you to everybody, and starting from the Quinnipiac, and thank you very much for organizing and hosting it and hosting the conference, to everybody who came here to listen to my presentation, and to our wonderful team who's actually doing the research and doing all the hard work to trying to make sure this is a reality. Thank you, everybody.
What's the inheritance pattern?
It's recessive.
So what does that mean in the family? Will you expect to see multiple cases?
Well, basically it's a 25% chance if both parents have the recessive gene. So if the two of the parents have the gene, it's with every kid it's a lottery. So it's a plus of 25% chance to get it. Yes, please.
You may have covered this, and if you did, I apologize for not paying enough attention. How long has MVID been known about?
From 1978. 1978. This is the first ever publication on the disease.
It's the first time I have ever heard of it, since 1978. And do you have any idea at all how many children might become affected with this?
So, right now there is 350 cases reported on the disease in total, starting from 1978 till 2007, about September 2017. 350 case reports. Some of them are duplicates. So the careful estimate and cautious estimate, I would say hundreds worldwide. But if we really consider the degree of the degree of diagnostic complexity, I would say possibly thousands.
Now, is that just within the United States, or is that worldwide?
No, global. Yeah, global.
Thank you.
Thank you so much.
Thank you.